Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Adoptive Cell Therapy Exploring and Influencing Heterogeneity of TILs and CAR T Cells. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speaker throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Samplex, who developed the content for this presentation. Samplex APS supports the life sciences and medical research communities with microfluidics-based solutions designed to deliver high-resolution insights into cells and genomes. Samplex stands ready to help academic, corporate, and government researchers reach their goals in areas as diverse as engineered cell therapy, molecular engineering, and cell biology. They have developed unique microfluidics instruments to support molecular engineering, Xdrops, and Xdrop sort. These instruments enable high throughput, sensitive, and accurate screening of functional population analysis with single-cell resolution by encapsulating living cells in highly stable double emulsion droplets. These picoliter-sized droplets act as microenvironments for incubation and screening. Xdrop can change how you analyze and work with cells. Now, it's my pleasure to introduce our speaker for today's webinar, and he is Thomas Hewlin. There he is. Hi there. He is a PhD student pursuing clinical cancer research in the group of Dr. Inga Marie Svan at the National Center for Cancer Immunotherapy at Haarlem Hospital. His PhD focuses on adoptive cell therapies using TILs and CAR T cells. Throughout his PhD, he has been involved in several projects and collaborations, including the initiation of Denmark's first academic CAR T clinical trial, DK-1901, an RNN-based CAR T production, and T-cell receptor discovery in unique tumor-reactive TIL populations. Now, before I pass the mic and the controls over to Thomas, we do have two poll questions for our audience to participate in. Now, this is done in real time, so your participation is strongly encouraged and very much appreciated. So please go ahead and click on any of the below that applies to you. Which of the following best describes you? Is it practicing medical doctor, cancer research or technician, scientific researcher other than cancer, student or non-scientific background? So please go ahead and make your selection. Oh, this is great, Thomas. It looks like we have the majority of our audience have participated in this poll, but I'm going to leave it open for a few more seconds just to get everybody involved here. So if you haven't made your selection, go ahead and do so now by just clicking on any of the options below. Oh, perfect. Thank you, everyone, for participating. Let's take a look at the results. So I'm going to close the polls now. Okay, so here's our results. In our audience, we have 4% practicing medical doctor, 43% of our audience is cancer researcher or technician, then 39% are scientific researchers other than cancer, 9% student, and 4% non-scientific background. So thank you everyone for participating in that poll question. And now we're going to jump to our second poll question. Another quick one, just 40 seconds we'll leave for this one here. I'm going to launch it now. And just like before, it's done in real time, so go right ahead and make your selection. What best describes why you attended today's talk? Is it because I'm interested in cell therapies for cancer treatment? I want to learn more about Xdrop technology? Or I joined for every X Talks? Okay, perfect. That looks like it's everyone. I'm so grateful for everyone's participation. I'm now going to close the polls and let's take a look at the results. Okay, here's our results. 78% said, I'm interested in cell therapies for cancer, while 22% I want to learn more, and 0% I joined for every X Talks. But I know you're out there joining us. All right, thank you very much, everyone, for participating in our poll question. And now I'm going to, I already shared the results there with you. And now let me hide that. And let me now pass the mic and the controls over to Thomas. So Thomas, when you're ready, you may begin.
Nice. Okay. Thank you very much for that, Sonia. It's good to see those poll results, to know who's in the audience today. Yes, so just to go over our agenda, first we'll just start with a quick introduction on cancer immunotherapy and cell therapy. We'll talk about heterogeneity. What do I mean? What diversity are we looking at? We'll talk about how we can influence TIL diversity and manipulate the tumor microenvironment. We will also talk about the Xdrop technology and the potential of it, how we used it to explore CAR T functionality and differences in potency. And we'll just conclude with some future perspectives and a discussion. Yes. So I'd like to begin just by posing the question, why would we use the immune system to treat cancer? So we already have multiple clinical interventions for cancer treatment. So some, such as chemotherapies, are very broad and a bit non-specific in how they target tumors. There are also very narrowly specific drugs like hormonal and targeted therapies, and these interfere with very specific pathogenic pathways that some tumor cells can rely on. There's also physical interventions like actually removing the cancerous or potentially cancerous cells, like surgery, bone marrow transplants. And we really just need to understand that among many definitions, cancer is a genetic disease and an evolutionary disease. And when cancer evolves, we need to alter clinical interventions that can adapt with it. So coming back to the initial question, why would we use the immune system? It turns out that actually the immune system changes and evolves in parallel with cancer. So some of us probably think of cancer therapy and immunotherapy as a relatively new field, but actually it's been almost 70 years since Frank Burnett and Thomas Lewis have made this tumor immune surveillance hypothesis. So they weren't really so much trying to put their finger on a definition for the immune system. They're mainly coming at it from the tumor and from the cancer perspective, right? So they realized that there must be some mechanism that's protecting us from neoplastic disease. And with every inheritable genetic change, it's a step towards malignancy. And it has to be an evolutionary necessity that we have these mechanisms for eliminating dangerous and mutant cells. And they reasoned that this must be of some immunological character. So they essentially dubbed the immune system as the natural predator of cancer. So it's not the perfect definition, but it really shows how the immune system can be so largely effective in the fight against cancer. And really it does this through its memory capabilities, the diverse cell types that the immune system has, the different effector functions that it also has, and its ability to discriminate between healthy cells and aberrant cells. So of course, it's kind of endless the way that the immune system interacts with cancer, but today we'll cover three different aspects. So today we'll talk about checkpoint therapy first, and to understand this therapy, we really have to understand immune tolerance. So I think the most succinct way to describe tolerance is in terms of infection. So say we have some infection, let's say a viral infection, it encounters the first line of defense of our immune system, so the innate response. So this is a very ephemeral response, and then it will hand things over to the adaptive system. So you'll notice here that it forms this overwhelming immune response to clear the infection from the host. And very importantly, it wanes. So it goes away after time, but it leaves behind some memory, just in case of reinfection. So this is exactly what we want. what we would not want would be, pardon me, an immune response that just continuously progresses unrestrained and causes constant inflammation and uh, everlasting response. So there's many ways in which immune tolerance works and maintains immune homeostasis. And one way it does this is through immunological checkpoints. So with this kind of tolerance, we're able to kind of dampen our immune response that we've generated. And two ways that this occurs is through the checkpoints PD-1 and CTLA-4. So when these receptors that are on immune cells interact with their natural ligands, they dampen this response, and we're able to kind of pull back the immune response. And this is really great when we're trying to fight a cold or some other infection, but it's really not what we want when we're trying to fight cancer. So cancer can actually exploit these mechanisms so that it can circumvent and escape the immune system. And in cancer, we don't want to dampen the immune response. We want to keep pushing it. So available now to patients are drugs called checkpoint inhibitors. And these drugs are antibodies, so small biologic molecules that can come and disrupt these interactions. And essentially doing this will cut the brakes from the immune response, assuming that the patient has mounted a pre-existing immune response, and hopefully overcome and fight the cancer. So moving from checkpoint therapy now to TILs, tumor infiltrating lymphocytes. While checkpoint inhibitors are off-the-shelf drugs, TIL therapy actually uses the patient's own immune cells. So how it works is through surgery, a piece or all of the tumor is removed from the individual. It's then taken to the clean room under sterile conditions and manually fragmented into very small pieces. Each one of these fragments, about 1 to 3 cubic millimeters, is put in a well with some stimulatory media. And then from these fragments, the young TILs or minimally cultured TILs can grow out from the media. So then we can harvest all of these immune cells and then put them into the rapid expansion protocol, or the REP. And this is done in a bioreactor like this. And it also contains all of these diverse proliferation signals that can be provided to the TILs here. So after expanding them to very high numbers, they can be infused into the patient with IL-2 so that we keep the immune cells alive. And the patient before this infusion is lymphodepleted, right? So these cells, we believe they're in the tumor for a reason. So number one, if they're in the tumor, they're likely more tumor reactive. And number two, they're obviously able to enter the tumor microenvironment, which is a barrier for other cell therapies. Yes. So now we can move to CAR T therapy, or chimeric antigen receptor T cells. Yes. So this statue is a depiction of a chimera in Greek mythology, and it is it's multiple species that are kind of combined into one hybrid animal that does not exist in nature, and that's similar to the CAR, which also incorporates a lot of different aspects of the immune system into one receptor. So earlier we talked about how the immune system can evolve with the cancer, and this is large part due to the T cells. So T cells are very picky though in how they can react to target cells. So they of course need to see peptide that is presented on MHC1 molecules. And not only that, they also need co-stimulatory molecules to activate them. So alternatively, antibodies can be engineered to bind to basically any molecular target. And they do that at their variable fragment here, the antigen binding domain. And so what a CAR T cell comprises is basically this antigen binding domain that's genetically linked to any of these co-stimulatory domains that you'd like to add. So you can kind of just stack them one on top of each other. So the most common target now for CAR T therapy in the clinic is CD19, and this is to target hematological malignancies. So the production for CAR T cells is very similar to TILs. Instead of taking the cells from a tumor, CAR T therapy removes T cells from the peripheral blood. And then to arm them with this CAR, with this receptor, the gene that encodes for the receptor is packaged into a virus that can then infect the T cells, insert this gene into the genome of the host cell, and then it can be expressed by the host cell. So then these are expanded, and then they can be infused back into the patient. So I'd like to make it clear that these are very different from any other drugs. So they're essentially made by the individual for the individual. So it's what we call an autologous therapy. So because every patient's cancer is different and every patient's immune system is different, this is how we get the word of the day, heterogeneity. So talking about these differences, we're going to narrow down to kind of the two main components of these therapies. So the receptors and also the cells themselves. So let's start with receptor diversity. So this is how the cells see and interact with the cancer. So in TIL therapy, we start with a TIL fragment, and that contains a lot of different immune cells with a lot of different specificities that we can illustrate with the different colors here. So when we expand these out of the fragment, what exactly are we expanding? What do we end up with? Do we end up with just kind of a copy, paste production of cells that exactly resembles what was in the tumor? Or do we maybe lose some of these populations, some of this diversity? Or it's also possible that maybe one or two or just a few of these clones kind of outpopulate all the other clones. And patients' TILs will typically fall somewhere within this spectrum of antigen diversity. So looking at CAR T cells, it's quite a bit more boring. So we arm them with one specific receptor that we know exactly the target that it will interact with, and it is this one clone that we expand. It's really just a question of how many of these cells express the receptor. So as of now, it's kind of a coin toss, whether we have a lot of knowledge of the receptor and low antigen specific heterogeneity, which would be CAR T therapy, or do we really not know a lot about the targets that the tumor and the TILs are interacting with, but we have a lot of heterogeneity within the populations. Yeah. So when we switch to phenotypic diversity, so this is when the CAR T cells can start looking a little bit more like the TILs. So yes, these are still T cells, but they can be quite different. For example, phenotypically. So what are the co-receptors that these cells express? Do they metabolize nutrients different or more the same? Do they have different reactive potential? If they do meet the antigen, are they able to react strongly as one another? And this is really, really in our interest to find out because they can have high impacts on patient outcome. So this graph here, this is from our phase 3 trial. It's a head-to-head comparison of TILs versus ipilimumab in metastatic melanoma. And this is in patients who progressed on first-line checkpoint therapy. So of course, we can see that the TIL group did much better than the IPI group here. And those that did have a response from TIL therapy had a very sustained response. But this begs the question, what happened to these patients that did not have a response? How do we reach these patients in this window here? So there are infinite possibilities why these patients progressed. And we have a lot of smart scientists and clinicians right now trying to figure out why. So then if we figure out what these differences are, what can we do to our TIL protocols? How can we intervene to improve our TIL therapy for patients? So we're already doing some things to try and improve the TILs that we make for patients. So for example, using CRISPR to knock out the PD-1 gene in the TILs. So hopefully kind kind of unexhausting or making exhaustion resistant TILs, which we hope are tumor reactive. But we also are curious, can we intervene at an earlier phase, maybe at this phase when the TILs are still in the fragment? Can we do anything to these cells so that we change the TIL output? So this leads us to this explorative study. So we essentially wanted to see, could we add checkpoint inhibitors to the TIL fragments and try and alter the tumor microenvironment, and then demonstrate some kind of plasticity in the TILs. So we added either anti-PD-1 or anti-CTLA-4 to these fragments, or no intervention and just IL-2 alone. So to assess this, we looked at cell number of the TILs that we got out, their reactivity, their phenotype, how they metabolize, and their antigen specificity. So we collected material from 15 melanoma patients that were already undergoing surgery to have TIL therapy. So one piece of the fragment was taken to the clean room to make their TILs for treatment, and another fragment was taken to the laboratory where we were able to do these experiments. So it's important to note that we have now essentially crossed this Rubicon of checkpoint therapy, and every metastatic melanoma patient, by the time they are eligible for TIL therapy, will have been exposed to checkpoint therapy. So this is an overview of the patients we treated. They all had some line of checkpoint immune therapy before this study, some more than others. And then this heat map just shows the unmodified TILs with nothing added to them, the TILs per fragment that we got out. So with the white lines here, you'll notice these are patients that we are unable to, at least in the lab, produce TILs from. So 73% of the fragments did produce established cultures, while these 4 did not fit the criteria for forming established cultures. So in these unmodified and just how we typically make TILs with these products, we wanted to look at some characterizations. So in the phenotyping, we found uniform terminal differentiation. Most of the TILs are of the T effector memory population. They all have a lot of variability in how much and which exhaustion markers they express. And also, very important, we wanted to see their reactivity when stimulated with tumor. So all of the patients did have a measurable reactivity to the tumor. So this is important, showing that patients that did receive 1 or even 3 lines of checkpoint therapy did produce functional TILs. So getting out of the way what we did not discover, we found that there was really no striking reactive or metabolic differences in the checkpoint modulated TILs. So when we introduced anti-CTLA-4 or PD-1, we see very similar levels of reactivity when stimulated with tumor, and there was not much change in the basal and maximum respiration of the cells, or the ATP turnover, or their dependence on glycolysis. So something that we did find that was interesting was that the PD-1 intervention was actually able to produce more TILs. And not just more TILs, but also in patients of fragments that we were unable to form established cultures, it actually was able to rescue production in all except for 1 of the fragments from these patients. So overall, what we see from PD-1 intervention is that we can kind of push more the proliferative capacity, to say. So with CTLA-4, an interesting finding was that it can broaden the scope of the reactivity that we see. So to explore this, we used this dextramer technology. So attached to this dextran backbone are MHC molecules with which we could load peptides from this library. So on one molecule, there is only 1 of these peptides loaded into all of these MHCs. They also have fluorochromes attached so that we can sort them through FACS. And then using this DNA barcode, we're able to amplicon sequence and then based on the unique barcode, trace it back to which of these peptides the cells are interacting with. So this heat map demonstrates our findings across the different conditions, and this can be summed up in these plots. So in the anti-CTLA-4 group, we notice that the number of specificities that the TILs can see, these are increased compared to no intervention, and also mildly increased are the sum of the estimated frequencies of these TILs. And then also what has been already shown is that in all conditions, these TILs reside in the CD39 and CD69 double positive population. Yes. So again, with anti-PD-1, we're kind of more affecting the cell's ability to expand. And with CTLA-4, we're influencing more on the receptor aspect. So yes, we can see some plasticity in the TILs and really influence the heterogeneity of what we can expand from the fragment. So now I would like to go back to CAR T therapy and just to introduce our trial that we began in 2022 at CCIT. So DK-CLIC 1901, this is Denmark's first academic CAR T clinical trial. It is simply just an anti-CD19 CAR with a 4-1BB co-stimulatory domain, a second generation CAR, and for production, we have a 12-day production protocol. So this is our production team. Everyone here is essential to both initiating the trial and also just its continuation throughout every single patient that we treat. Yes, so this is just an overview of the first 5 patients. We do in-process monitoring on day 5, day 8, and day 12. So here we can see the number total of CAR-positive cells. You'll notice a lot of differences between the patients here, how many cells are produced. And then the bottom graph here, we can see the CAR expression across the days and across the patients, and within the CAR expression, the CD4 and CD8 makeup of the CAR-positive cells. Yes. So with these cells, I do a lot of killing assays, mostly this conventional bulk killing assay, so chromium-51 assay. And here's an example from our first patient of the trial. So they were infused with an infusion product that is 36% CAR positive. So the target cells here are the Dowdy cell line. So this is a CD19 positive cell line. So the CAR T cells can interact with them and hopefully kill them. The K562, these are a CD19 negative cell line, so they should not be killed by the CAR T cells. So we see exactly what we would expect here, killing in the Dowdy populations. But which cells in the infusion product are doing it? Well, of course, we would all say it is the CAR positive cells. They're doing the bulk of the killing. Now, we also have a validation product. This is a pure CAR culture. So all of these cells here are CAR positive. And so, of course, we see more killing. But now we have to ask, is every single CAR T cell doing the same amount of killing and equally contributing to the assay? So we would likely say no. And this is because these cells are different. There's a lot of heterogeneity in this population of cells that is 100% CAR positive. So if we look, there are some CD4 cells, there's some CD8 cells. If we look at cytokine production, a lot of them are double positive for producing cytotoxic cytokines, but some of them are single positive. And the same is true for these activation markers. So this makes us wonder, what could be the difference with these cells? Which ones are doing the bulk of the killing in this assay? So maybe it's the cells that have more CAR than the ones that have less CAR expression. Maybe it's one phenotype. Maybe it's the CD8 CAR T cells that are doing the bulk of the killing. Or maybe it is the expression of a particular gene that allows the cells to be more potent. Or if it's not a gene, maybe it's a pathway that has that's giving the cells more proliferative potential. And so this nicely brings us to what we can do with the Xdrop technology. So just to first show kind of the anatomy of the droplet and what exactly we're talking about here, the cell can actually be encapsulated into this bilayer where it can exchange nutrients, but it still contains all the secreted chemicals. And they just sit here with medium on the inside and on the outside of the droplet. So the structure and the function of the technology is quite intuitive. So in this cartridge, all of the reagents will be loaded. There will be this insert where you can add up to 2 different cell types, so one on each side of this divider. And then the cells will be pulled in through the microfluidic system. They'll pass through an oil phase and then an outer phase mix. And then what will come out are these very uniform droplets, as you can see from the images here. So once these droplets are formed, it's really up to the user what they want to do with them. So you can do flow cytometry and cell sorting because these droplets are compatible with just basic flow cytometers that we likely all have sitting in our labs. You can do microscopic imaging, and then you can also rupture the droplets and harvest the cells afterwards. Yes, so now I'll show some experiments that we've done with these and CAR T cells and TILs, and all of this work was done with the help of my colleagues, Chris and Anna Christine. So to demonstrate what we can do with the Xdrop, we have 3 main assays that we've done. So effector and target cytotoxicity assays, cytokine secretion assays, and post-cell assay recovery. And the really nice thing is it's not just one of these that you can do. You can really integrate any combination of these assays. So this is just an effector target cytotoxicity experiment that we did. So for the setup here, we have either 100% CAR T cells or non-transduced cells, so CAR negative cells. These are our effector cells and a CD19 positive target cell line. So after staining the cells with 2 different colors, they go into each side of the insert here. We also add a dead cell stain, so propydium iodide will detect dead cells. And then we run the protocol, and then we harvest the production. So what comes out of the cartridge is this suspension. So we have the droplets that come down to the bottom, and then they sit with media in the cells, and you can just incubate these at 37 degrees, just like they are regular cells. Yes, and then so we took these over a various time course and ran them on our flow cytometer. Yes, so once they're ran, when you look at the side scatter, forward scatter, we get this large population here, and these are the droplets. So you can gate out these droplets, and then since we have 2 parameters here, you can make these quadrants. So in these quadrants, we have at the bottom, the double negative is going to be the empty droplets. So there are a few of those, and then we have also double positive. So this is what we call the co-encapsulation. So this has 1 of each cell type. And then, of course, we have single encapsulations, so the target cell only and the effector cell only. So within each of these quadrants, this is when we can look at the PI channel. We can look at the cell killing based on the propydium iodide staining. So in the empty cells, of course, we see none. There's some background death in the target and effector only cells. And then we see a lot more killing in the co-encapsulation populations. Yes, so we did this both with CAR T cells and with TILs, and we did time course studies on both of them. So here, like I introduced before, we have CAR T cells, pure CAR T, and then we also have non-transduced CAR negative cells. So we can see the killing over time with those. And then also with the CAR T cells, we have autologous TILs versus wild type tumor. So the TILs and the tumor have been derived from the same patient. So we see our increased killing over time with that. And then we also have a knockout of the MHC gene, so they cannot see antigen, so that's why we do not see any killing here. And then we also have allogenic TILs with wild-type tumor, so tumor from a different patient that they cannot interact with. Yes. So going further, we're able to do the same assay, but actually integrate in the cytokine secretion assay. And the cytokine we are interested in is granzyme B. So CAR T cells, when they're activated, they will produce granzyme B to kill the target cells. And in this assay, when granzyme B is produced, it will trigger a fluorescence. And this we can quantify so that we can measure killing. So again, we have here our 4 quadrants. And if we look at our single encapsulations, we have very low background through the FITS channel, which is how we are detecting the granzyme B, and same for the CAR T. Now looking at our co-encapsulation quadrant, here we have in the CAR T positive data set over time, very nice increased granzyme B production, and then some increase in the non-transduced cell as well, but much lower. Yes, and then so we can put this into a graph so that we can see the comparison. Yes. And then looking at the time course and all of our parameters here, we're able to see that in the CAR T population, we have this increasing granzyme B population, and then eventually that population will spill into the cell death population here. So as the cells produce granzyme B, then the target cells will start to die as a result. So we do see this pretty consistent cell death population. And our theory to that is that these CAR T cells, they just kill very quickly. And that is one aspect. And another aspect is T cells, they're very diverse in how they respond. So there are many other ways other than just granzyme B secretion that they can kill target cells. Yes. So from these, again, we can make these bar graphs for comparison. And then also in this experiment, we're able to actually take the productions and put them under the microscope. And if you have a fluorescence microscope like this, and you can detect these channels of your stains, then you can visualize the droplets like this. So if we look at this like really just textbook image of the droplets, you can actually see the 2 cells, the very small CAR T cell in the purple, and then the very large Dowdy, the tumor cell here. And then you see that it is also stained for the propydium iodide, so for cell death, and we have a lot of granzyme B production here. But looking at kind of all of these droplets, we notice that some are very bright, and these are really high granzyme B producing cells, and some of them are very low. And so these are not producing much granzyme B, even though they're co-encapsulations. So of course, something is different about these cells, since they're both CAR T cells. So from here, what we could do, which we plan to do, are we can sort these droplets based on granzyme B production, and then we can actually rupture these droplets and recover the cells, and then do some more downstream analysis to find out the differences between these cells. Yes, so then to go over another assay, using NK cells this time, we can demonstrate how you can integrate the cytokine secretion with the post-assay cell recovery. And we just used a commercial Miltenyi interferon gamma capture kit for this. So NK cells were chemically stimulated to become activated. The interferon gamma capture kit was used. The Xdrop protocol was ran for encapsulation, and then they were ran on the flow cytometer. So here we see a strong interferon gamma positive population. So from here, we're able to do some FACS sorting and then post-assay cell recovery. These cells were then put into culture for 2 weeks, just let sit, and then re-stimulated. So the green histogram here on top, we see the selected and reactivated NK cells. We can see how they change to being a mostly interferon positive population. And then the purple graph here, we have the non-activated cells. And then some other NK cells used as a control that are activated and non-activated. So we can show that we're able to activate the cells, sort them, rupture the droplets, and culture the cells, and they are able to retain their activation phenotype. So really, we're moving now from these bulk, low-resolution assays to being able to do these integrative and multi-parameter, very high-resolution assays. And it's here that we can really distinguish and explore the heterogeneity in all of these cell populations. So I'd like to just kind of end today with one of my favorite quotes from one of my favorite books. So all animals are created equal, but some animals are more equal than others. And we can kind of say the same about cell therapies. So it's 2024. Thousands of individuals have gotten cell therapies, so CAR T cells and TILs. And to an extent, these are really only the same therapy by name. So given all the heterogeneity that we see between them and the enormous diversity that we discussed. And now we're at a point where we can really shape this diversity to hopefully have some kind of clinical impact and also precisely observe it at a very fine level. So with the Xdrop, we're able to really elucidate heterogeneity in cellular populations and really assess the effector functions of these cells. And we hope that we can identify very meaningful differences in these populations. And then maybe we can apply these discoveries to influence patient outcome. So with that, I would just like to really thank a lot of people. So definitely Samplix, thank you very much for giving me the opportunity to present this work. Thank you to my colleagues at CCIT who helped out with all of this. Also DTU Health Tech. These are my affiliations and funders. And with that, I will pass the mic and the controls back to Sonia. Thank you very much.
Well, first, let's say thank you very much, Thomas, for that very engaging presentation. I hope everyone enjoyed it. And now we're going to begin the Q&A portion of the webinar. So I'll invite Thomas to put on his webcam. Hi, everyone. And just inform our audience that you still have the opportunity right now to send in your questions using the questions window. And we'll try to attend to your questions during the time that we have together. So while you were speaking, I did receive a bunch of questions. So let's start with this first one here, Thomas. This audience member is asking, you mentioned that CAR T cells don't have antigen specificity, heterogeneity. Do they still have indigenous TCRs?
Yes, so we have to remember they are still T cells, so they do have endogenous TCRs. We don't think that it is the main driver of how potent they are against cancer. So really, it's the CAR that is doing most of the heavy lifting in the tumor reactivity. But yes, they do have endogenous T cell heterogeneity. Yeah.
Okay, perfect. Thank you for that, Thomas. I'm just getting through the questions here. Let's go to this one here. This audience member is asking, is the droplet production protocol different depending on the experiment? So they're saying, in other words, are droplets made with the same reagents for all experiments, or do different droplet kits need to be purchased depending on the experiment? That's a very good question. What are your thoughts on that, Thomas?
Yes, so the production of the droplets is the same every time. It is standard reagents provided by Samplix, but outside of that, you can integrate pretty much any commercially available stainings or reagents you want to do. So if you want to stain for particular markers, or whether you want to have some kind of other reagent like the granzyme B reagent, which is not an antibody marker, you can buy these commercially, but they will be used all with the same kit to produce the cells.
Okay, all right, thank you for that explanation. We'll go to the next question here. This audience member is asking, can you speak to the cell viability when encapsulated in the droplets?
Yes. So, um, like I mentioned in the presentation, you can just put the productions with the media into the incubator, and
Mhm.
you essentially just treat them like they are cells. And in doing that, you can do a time course assay. We went to 24 hours with the TILs.
Mhm.
So from personal experience, we know that we can still do experiments, still have viable cells after 24 hours. Samplex may have gone further than that, I'm sure, to test how far they can push the viability in the cells, but definitely up to 24 hours in our personal experience.
Okay, thank you for that. Let's jump to the next question here. This audience member is asking, are the cell types discussed today the only cell types that have been tested using the technology?
In our experience, we've tested the CAR T cells and the TILs, also NK cells. These are the three that we personally have experience with, but I know that Samplex they've even tested it in mouse cells in more in splenocytes. Um, so yes, I think even beyond that, a lot different cell types have been tested.
Okay, all right. Uh, this audience member is asking, in the first study, all patients included were previously treated with checkpoint can I say that probably? Inhibition therapy. Do you think this influenced your experiments at all?
I have to say yes. I think that I think well we what we do know is that at least with TIL therapy, uh, previous exposure to immune therapies, these patients can produce uh, reactive TILs, um, but it's not quite as potent. So I have to think that yes, given that these patients probably already had a presence of checkpoint inhibitors uh,
Mhm.
interacting with their immune system. This probably influenced our experiment when we tried to, again, put checkpoint inhibitors into the tumor microenvironment. So yeah,
Mhm.
I can't say how it affected it, but I would assume, yes, it definitely did.
Okay, thank you for that detailed answer. Okay, I think we have time for a couple more questions. And this is just a reminder to our audience members, you still have the opportunity to send your questions in, and we'll try to attend to them during the time we have together. So let's just go to this next question. Why are patients lymphodepleted with TIL and CAR T therapy? What are your thoughts on that?
Yes. So with these adoptive cell therapies, um, we're essentially trying to replace their immune system with an immune system that is tumor reactive. So we give the chemolymphodepletion to essentially wipe out their pre-existing immunity so that we can replace it with the CAR T immunity or the TIL immunity. And also, in doing that, we are able to also deplete kind of the suppressive immune cells that the patient has, so that we can really drive the cancer specific response.
Mhm. Okay, thank you for that. Okay, we'll go to the next question here. What flow cytometer did you use in the Xdrop experiments? Do they different optimizations need to be done for each cytometer?
Yeah, so we here use a Novocyte Quantion, um, and then at Samplex they have a Sony cell sorter. I don't remember the model, um, but uh, there's some adjustments and optimizations that need to be made. So the voltages need to be optimized, and then also the compensations. David at Samplex is really good at optimizing the compensations so that you have really nice compensations when you
Mhm.
run the droplets. So yes, some need to be done, but Samplex is really good at making sure that you can optimize those for your experiments. And yes, you can in there use any commercial flow cytometer.
Okay, well, thank you very much, Thomas, for answering all those questions. That's it for our Q&A portion of the webinar. For those of you who are interested in hearing more from Samplex, save this date, June the 18th. They have another webinar scheduled. When the information becomes available, we will send it to you, and then you'll be able to register for another exciting webinar. Now, if we were not able to attend to all your questions, remember the team at Samplex and Thomas will follow up with you after this presentation. And if you have any further questions, please direct them to the email addresses that you see there on your screen. Now, give me a few seconds. I'm going to share the recording of today's event in the chat box, which you can also share with your colleagues so they may register for the recording here as well. And a survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. So I encourage you to do that. Now, please join us in thanking our speaker, Thomas, for his time here today. So thank you very much, Thomas. We appreciate your time.
Thank you. Happy to be here.
Okay. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at Xtalks, we thank you for joining us. Until next time, I'm Sonia Hunt. Please take care and bye for now. Bye, everyone. Bye, Thomas.